in the
Organisms.
whole organism. Okay. Know the difference in your four major macromolecules. So carbohydrates are what?
Energy?
It is, sure. So there's your polysaccharides that are broken down into individual monosaccharides. How about proteins? They're long chains of
Amino acids?
amino acids. What about lipids? What are those?
Fats.
Fats and oils, which are fatty acids and glycerol. Yep. And the nucleic acid, what is the unit? One unit. It's made up of a 5-carbon sugar, phosphate group, and the nitrogenous bases. Remember, that's your A's and T's and C's and G's that join together in DNA. You know, it's the double helix, so the rungs of the ladder of the DNA. That is the nitrogenous bases. ATP also is a nucleic acid. So DNA, RNA, ATP. Make sure you know all the parts of the cell, what's the function of the plasma membrane. Yes, it controls its the permeability, what is able to go in or out of that cell. It is a barrier, so it holds everything in, it keeps stuff out. What's it made up of?
Phospholipid
The
bilayer.
phospholipid bilayer.
Just do some
What is this? Villi.
What is this?
memory.
The bicuspid.
That's the
Microvilli.
microvilli. Oh, what else is in that phospholipid bilayer? What's kind of shoved in there? Proteins that allow what?
So like a transport.
Yeah, so specific things going through, like based on size or based on charge, stuff like that. Cholesterol's another thing that's shoved in there. So the plasma membrane is going to regulate what goes through. What if it does use that phospholipid bilayer no? What if it uses the proteins in that phospholipid bilayer to go through? What is that called? No. Facilitated diffusion. The act of transport would be when it's going from a low concentration to a high concentration. So against the concentration gradient. And yes, just like that sodium potassium pump, it is using a pump, but if it's going from a low concentration to a high concentration, it's gonna make it even a higher concentration on the other side. So you have to have energy, ATP, to force it in that direction. How about the nucleus? What's in the nucleus? The DNA, so the genetic material. What is it called when that DNA unzips and makes that single-stranded copy?
Transcription.
Transcription is first. And the messenger RNA then goes to what organelle in the cytoplasm?
The mitochondria? No, the ribosome.
Ribosome. Okay, that messenger RNA zips through that ribosome to make what? Proteins. Protein synthesis, that is called what?
Translation.
Yes, translation.
My head's burning.
What is the endoplasmic reticulum?
Pathway.
That's right. That's this. That is the roadway of the cell. I'm so glad though. Like, see, it helps. It helps. It's the roadway of the cell. And if it is rough, that means it's studded with ribosomes that are making proteins. So those proteins are being transported across the cell, usually to the what?
Golgi apparatus.
Golgi apparatus, which does what?
Packages.
Sorts
UPS.
and packages, UPS sorts and packages and prepares for export. How about lysosomes, what are they?
They're the,
Yes, the stomach of the cell, so they have digestive enzymes in it to help get rid of waste.
Pac-Man.
That's my macrophages.
Oh.
What about mitochondria?
Powerhouse.
What do they make?
ATP.
ATP, what is that called?
Adenosine triphosphate.
Yeah. What is the production of ATP called?
Oh.
Cellular
That
respiration,
part.
right? And what do you have to have for that?
Glucose.
Starts with glucose. Yep, and then it's converted to pyruvate and then ATP. So the difference in active and passive transport, active
Required energy.
requires ATP because you are forcing something against the concentration gradient. Simple diffusion or passive transport, simple diffusion is an example of that, goes from what concentration to what?
High to low.
High to low, in order to reach
Equilibrium.
equilibrium. Okay. What's the difference in mitosis and meiosis? Mitosis is for what?
Cellular replication.
Yeah, sure. Okay. So that is
has
just, that's
all the-
regular division, that is
Yeah.
for growth, repair, specialization, all of that. And how many chromosomes?
The same
The cells are identical, so you end up with how many cells? 2 cells, 46 chromosomes in each. Meiosis is for the production of gametes, sex cells. So those are what?
They only have half.
They do have half, which is how many chromosomes? 23. That's right. And what are those 2 gametes? What is this?
Sperm and egg.
What's this called?
Fertilization.
Fertilization. Y'all are so good. We should just play charades for our Bible exam.
Right, you're all right.
I know, you'll be doing good.
Just have little pictures on the, by each question. The hand
Okay, what
in
is
there.
What is it called if it only has 23 chromosomes?
Zygote? No.
The ploidy, what is the ploidy? If it only has 23, it is haploid. You never heard that word? Haploid. Hap is half the normal number. And what is the 46 chromosome called? Diploid, yes. Di, that's 2. That means that you have the pairs, right? And you get one of each of those pairs from mom and one from dad, right? So you get one of the number 1 from mom, one of the number 1 from dad, all the way down to the 23rd pair, which is your sex chromosomes. If you're XX, you're what?
Female.
Female. If you're XY, you're male. Okay, the different types of tissues. Epithelial tissue is what? Covering and lining membranes. Covering and lining. It is avascular, meaning that there's no blood flow going through it. And those are named based on the number of layers. Simple is
One layer.
1 layer, stratified is
Multiple.
more than 1. And the shape, so if they're flat,
Squamous.
squamous, if they're square, cuboidal, if they're tall, skinny, columnar. What is this one that the cells start to spread out like this?
Transitional.
Transitional, and that allows for
Stretching.
what about the ones where they're tall and skinny, but they're so squished together that it almost looks like stratified, so they're kind of like this?
Pseudo-stratified.
Pseudo-stratified, and sometimes they have what on top?
Villi.
Cilia.
Got that.
Say, villi, cilia.
Villa and then Celia.
They look the same.
My mom's name is Celia, so I can
Your mom had a cilia.
like Celia. C-E-L-I.
That's cool. I've never heard of that.
I don't know where my grandparents go.
You could just call her this.
I know.
Hey, Mom. Okay, what else? If it is simple as one cell layer thick, so that's perfect for what?
Diffusion.
Diffusion, yes. Diffusion, so things go through it very easily. So simple diffusion.
Easy like me.
What else do we have? We did say facilitated diffusion is using
Protein.
the protein carrier molecules, yes. What is one that's based on size and pressure? What's that called? Filtration. What is, where do you find simple squamous?
Alveoli.
Alveoli, what's surrounding those?
Capillary.
Capillary, what is this?
One cell layer thick.
Yep, and they're flat.
Oh yeah, squamous.
Simple squamous, this is simple squamous, right? So alveoli, capillaries, the glomerulus in the kidney, because that's just capillary, not. Okay, where do you find simple cuboidal? What is this?
Digestive tract? No.
What is
No.
this? Twisty turny like this.
Intestines.
Convoluted tubules.
The convoluted tubules in the kidneys. So those are simple cuboidal, which are surrounded by the peritubular capillaries, which are simple squamous, both one cell layer thick, so that's perfect for what?
Reabsorption.
Reabsorption, okay, and then secretion at the very end. What is one layer tall and skinny?
Simple columnar.
Where do you find that?
Digestive tract.
Digestive tract, which is perfect for
Absorption.
absorption of nutrients, right? And what's just on the other side of what's inside the
Villi.
villi? What's inside the villi? That's absorbing.
Oh.
Capillaries. The capillary loop inside those villi is absorbing all those nutrients that are the smallest, right? Like amino acids, individual sugars, stuff like that. So it is absorbed into the villi, into the capillary blood, taken to the liver via the hepatic portal vein for processing of nutrients, right?
Digestive system. I'm gonna brush up.
Connective tissue.
Blood.
Adipose is
Fat.
adipose tissue.
Fat tissue.
That's fat cells. What does it look like?
Bubbles.
Big bubbles.
This chair. And it looks like the signet ring appearance, remember, with the nucleus pushed over to the side because of that giant lipid droplet that's in there. Remember, they shrink when you start to lose weight. Areolar. It's like a packaging, right? Looks like twigs and berries. Reticular, you find that in your lymph organs, so there's a lot of immune tissue in there. Blood, what's blood made up of? Red blood cells, white blood cells, platelets, plasma. Excuse me. It is the connective tissue that has the liquid matrix. How about bone? That's connective tissue. We have compact bone, we have spongy bone. The compact bone is made up of all those circles called osteons, all those concentric circles. It has the central canal, reversing canal in the middle, and then Volkmann's canals that connect those. And it has the hard calcified matrix. How about cartilages? Elastic cartilage, where do you find it?
Epiglottis and ears.
Ear and epiglottis, where do you find fibrocartilage? Disc. So pubic symphysis disc, meniscus disc, and intervertebral disc. Everything else is hyaline. Okay, where you find hyaline? the trachea, the larynx, the costal cartilage, the ends of long bones, which we call articular cartilage, the entire embryonic skeleton. Okay, what else? How about dense, regular connective tissue? Tendons, ligaments, aponeuroses, very, very strong. How about dense, irregular? Capsules surrounding joints, because you get a pull in many directions with that one. Muscle tissue, what do you have? Skeletal, which is voluntary, striated, multinucleated. Cardiac is striated, involuntary, uninucleated, branching, intercalated disc. Smooth is not striated, involuntary, spindle shaped. Okay, and nervous tissue is what? Neurons and neuroglia. The neurons are the ones that send the electrical impulses. Neuroglia are the helper cells. We've mentioned those, like astrocytes cross the blood-brain barrier. Ependymal cells secrete cerebrospinal fluid and circulate that. Schwann cells make the myelin sheath in the peripheral nervous system. Oligodendrocytes make the myelin in the central nervous system. Satellite cells surround cell bodies in the peripheral nervous system. Am I missing any? I don't think of. Okay, and we talked about neurons sending the electrical impulse. Okay, what is hypertrophy? An increase in what? Cell size. How about hyperplasia?
Wasting away.
Number of cells. How about neoplasia? What's a neoplasm? It's like a tumor, so that's uncontrolled growth. And neo means new. What is metaplasia? That's a change from one tissue type to another. And what is atrophy?
Wasting away.
Yes, a decrease in cell size from tissues usually. All right, tell me about your vitamins. A and C are for what? Skin, immunity, collagen production. Vitamin D, what you have that for?
Bone.
Calcium absorption, absolutely. What about vitamin B? Red blood cell formation is a big one. E is anti-inflammatory. It's also good for wound healing. What does hydroxyapatite do? It hardens the bone matrix. Okay. How do wounds heal? If it looks exactly the same, it's called regeneration. If it is scar tissue, that's called fibrosis. A first-degree burn is only what?
On that, what
Gosh, you
Steven's
are
got
ready.
right now.
Steven.
I got burnt.
Goodness,
So I know
that's
what it
first
is.
degree, so it is red and painful. You have damaged your epidermis.
All over.
What about second-degree burns?
Blisters.
How many layers?
2.
2 layers. So you've destroyed the epidermis, damaged the dermis. That's where you find blisters. Third degree, how many layers?
3.
3 layers. You have destroyed the epidermis and dermis, damaged the hypodermis, which is made of what? Adipose tissue. And third degree burns, do they hurt?
No.
No, because you destroyed the nerve tissue. Sometimes it is like white, or it can be black and charred. And what do you have to worry about with that?
Infection.
Catastrophic fluid loss and an infection. Yeah, both of those. And what? Electrolyte imbalance. because you lose electrolytes
Fluid,
with
yeah.
the fluid, so that can make a big deal too. What else with skin? So, what are the organelles what are the organs in the skin? So, we have the hair follicles, we have the nerves going to the hair follicles, we have Pacinian corpuscles, which are
Touch.
deep
Pressure.
pressure receptors. You have the Meissner's corpuscles, which are light touch receptors. You have free nerve endings, which are touch, vibration, pain, temperature. Okay. You have erector pili muscles, which do what?
Make the hair stand up.
When you're
Cold.
cold or scared. What else is in there? Sebaceous glands secrete
Oil.
oil into the hair follicle. Eccrine sweat glands secrete sweat to a pore in the surface. Okay, they have the long duct. Apocrine sweat glands produce the milky white, and that is dumped into hair follicles in the axillary and pubic region. Those are your two types of sweat glands. Okay, anything else with skin? The two parts of the dermis are the papillary layer, which is the top, that is kind of wavy. That's where you find the Meissner's corpuscles and the capillary loops that are taking oxygen to the epidermis because it's avascular and getting rid of carbon dioxide and waste. And then you have the reticular layer at the bottom. Okay. Bone, osteoblast,
Create.
building, osteocytes.
Destroy.
No, site. Site is regular cell.
Okay.
So osteocyte is a mature bone cell. Osteoclast,
Destroy.
they break down the bone, so they destroy the bone, and they leave that indentation behind them called what? The resorption bay, and it looks kind of like a slug that leaves its little mucus trail. Remember, they're the scrubbing bubbles, osteoclast. What else about bone? So compact bone is made up of the osteons, we talked about that. Spongy bone has space in between because it's just like a sponge. What do you find in that space? Red bone marrow. And what is that for? Blood cell formation. The bones, so long bones have a top, proximally epiphysis bottom, distally epiphysis, diaphysis in the middle. What's in the middle of the diaphysis?
Marrow.
Yellow bone marrow that is made up of fat. I guess that's good on bone. Muscle, what is this? That is, you're close. This is transitional. Okay, they spread out and gets thinner. This is the sliding filament theory. This is my actin and myosin, right? That is called the sliding filament theory, where the thick myofilament myosin heads grab onto the thin and pull it towards the line in the middle, which is called the M line. The H zone is just myosin, where the actin is not overlapping it yet. So as it contracts, the H zone gets smaller. Actin is attached to the Z disc. The myosin is attached to the M line. What is the functional unit? We have that one model. What was it called? That microscopic muscle model? Sarcomere model. What was surrounding it on the outside? Sarcoplasmic reticulum that stores calcium. What is the three things together? 3, triad. Remember, the electrical impulse goes down the T tubule, transverse tubule. That comes into contact with the terminal cisterning of the sarcoplasmic reticulum. So when that electricity goes down the middle, it opens up those calcium voltage gated channels and allows the calcium to flood out and go deep, right? Where's calcium bind? To troponin. And it changes shapes and moves. Tropomyosin off the myosin binding site so the myosin heads can attach to actin, pull it in. When it's attached, what's it called? The cross bridge. When it pulls it in towards the end line, that's called the power stroke. What is the motor unit? One motor neuron and all the muscle fibers that it innervates. Okay, so if we want a stronger contraction, we're going to incorporate more motor units. What is isotonic? Contraction and shortening. Which one is contraction with no shortening, like this? Isometric. Isometric, contraction with no shortening. Isotonic is contraction with shortening. What's the difference in aerobic and anaerobic?
Oxygen.
Which one?
Anaerobic has no oxygen.
Right, because an means not. So anaerobic, no oxygen, aerobic with oxygen. If you have no oxygen, like you're working out really hard, and you're compressing those vessels, you're not getting the oxygen to the muscle that's needed. So it is going to switch from regular cellular respiration to what? Anaerobic, well, it's not called respiration, but anaerobic, which is, what is it called? Fermentation. Fermentation, and in humans, that fermentation causes the production of what that makes you sore? Lactic acid. Yeah, lactic acid, and that's when it's anaerobic, no oxygen, so it switches to lactic acid fermentation. We talked about the differences in the muscle types. We talked about the different neuroglias. Oh, I didn't say microglia. Those are the immune cells. I mentioned the other ones. Oh, how about that graph that I drew? Resting membrane potential. What is that where it normally stays at? What number? -70. -70 is resting membrane potential. When, let's just start from the very beginning. I'll do it one time. Okay, that action potential comes down the axon of the motor neuron to the axon terminals. That electricity causes the calcium voltage gated channels to open. Calcium floods into the axon terminals, which makes the synaptic vesicles release the neurotransmitter, which is called acetylcholine. Across the synaptic cleft, that acetylcholine is going to bind to the receptors and open them up. What floods in? Sodium, which is what charged? Positively charged. So when the positive starts to go in, it goes up from the -70 towards the positive, and if it hits -55, what is that?
It's the
Threshold. Threshold. And threshold, that means all or nothing. Either you reach threshold, you get the depolarization where it spikes all the way up to what?
Positive.
30. All the way to +30. What is that called?
Polarization.
Depolarization. Y'all better look at that graph again. Depolarization, it goes all the way to positive 30.
So that's just
So if it
opposite.
reaches the threshold, -55, remember all the other sodium channels open and you get that massive influx of sodium, which is positive. So it makes that -70 go all the way up to the positive 30. When you get there, sodium channels close, potassium channels open. Potassium's going out. So potassium goes out, that is called repolarization. So it goes back down to the -70, a little bit under, that is called hyperpolarization. Okay, make sure you know that.
If you don't reach it, you get muscle failure. I know that.
Why is it negative on the inside? Because you have a lot of negatively charged proteins, right? It's positive charged on the outside because you have tons and tons and tons and tons and tons of what on the outside? Sodium. Okay, sodium. So it's pretty easy for that sodium to come in, right? So it's positive on the outside because of the sodium. So that makes it polarized, negative on the inside, positive on the outside. And that polarization is what causes all the contractions and the impulses moving across neurons, the axons and stuff, okay? Since we took a whole bunch of sodium in and spit the potassium out, we have to replace that, so we use what to do that? The sodium potassium pump, okay? And remember, that is active transport because we have to pump the sodium back out, but that's going from a low concentration to a high concentration, remember, because you have so much sodium on the outside. And then bringing that potassium back in, so that is active transport, replacing where they went after the depolarization and repolarization. What definitely do you need for the nervous system? The sodium and potassium, okay? What is an action potential?
That's the signal moving, right?
Yeah, that's that electrical signal. Let me finish that. So I had the neurotransmitter, which was acetylcholine, come out and move across that synaptic cleft, bind to the receptors, and then they open up and allow the sodium to come in, right? So when that sodium comes in, if you reach threshold, all the other sodium channels open and you get that massive influx of sodium, right? So that is then going to send the electrical impulse down that T-tubule, which causes the calcium voltage gated channels in the terminal cisternae to open up and allow the calcium from inside the sarcoplasmic reticulum to flood out. It goes deep inside the muscle, it binds to troponin, troponin changes shapes, moves tropomyosin off of the myosin binding sites, so now the myosin heads can attach to the actin. Now what do you have to have for the myosin heads? ATP. Okay, so when ATP binds to it, it breaks down into ADP, an inorganic phosphate. It goes into the COP position. Phosphate comes off, it attaches to actin, forming a cross bridge. The ADP comes off, you get the power stroke. When another ATP binds to the myosin head, it detaches, and then the whole thing happens all over again. So what happens after you die? You run out of what? ATP. So the myosin heads don't detach, so you stay tight, which is called rigor mortis. Okay, good. So we talked about the endocrine system and the hormones. What is the target tissue? It's what those hormones go to tell what to do. So the target tissue of the hypothalamus is going to be the anterior pituitary, okay? Because the hypothalamus produces those releasing and inhibiting hormones that pretty much tell the anterior pituitary whether or not to release those hormones. The hypothalamus, remember, also makes oxytocin, which is the love hormone, antidiuretic hormone, which goes to the kidneys, tells them to reabsorb more water. But those are secreted by the posterior pituitary. Okay, what are the anterior pituitary hormones? Growth hormone, cellular growth, but especially muscle and long bone. Prolactin causes milk production and release. Those are the two that are not tropic hormones. The other four are tropic hormones, meaning what? They go to other glands to tell them what to do. So thyroid stimulating hormone goes to the thyroid, tells it to release thyroid hormone. Adrenocorticotropic hormone goes to the adrenal cortex. Luteinizing hormone and follicle stimulating hormone go to the gonads. Follicle stimulating hormone tells the follicle to grow and mature. Luteinizing hormone also controls the hormone release from the gonads. Then we have thyroid gland. Thyroid hormone controls metabolism. Calcitonin does what? Decreases blood calcium levels. It's released when you have high calcium levels. So it will deposit it in bone. Then we have parathyroid gland that secretes parathyroid hormone that is released when there's low blood calcium. So it steals it from the bone by stimulating osteoclast. It breaks down the bone, releases that calcium into the bloodstream, making the blood calcium levels go back up. Pancreas secretes insulin when you have high blood glucose levels, and its purpose is to decrease those blood glucose levels and make the glucose go into the cells where it's going to be needed for cellular respiration. Glucagon is what is released when you've been fasting. So it is going to increase blood calcium levels because it's going to tell the liver to break down the glycogen stores and release that glucose into the blood. Okay, the adrenal glands, aldosterone, controls sodium balance, cortisol, cortisone, corticosterone, all those are your steroid hormones. Cortisol is the stress hormone, and then you have androgens. All those are from the adrenal cortex. The adrenal medulla secretes what? Epinephrine and norepinephrine, which is adrenaline, yep, fight or flight, sympathetic nervous system causes the secretion of that. Then you have the testes secrete testosterone, and the ovaries secrete estrogen and progesterone. Estrogen, secondary sex characteristics, progesterone keeps the uterus at bay, not contracting. Okay, and both of those are secreted by the corpus luteum after ovulation. Is there anything else? Any other glands? I can't think of any more. The thymus. Yes, the thymus helps mature T lymphocytes, so it is also part of the lymphatic and immune system. Thyroid stimulating hormone, its target tissue is what? The thyroid gland. And follicle stimulating and luteinizing hormone, their target tissues, the gonads, adrenocorticotropic hormone, the adrenal cortex is its target. So remember, those are going to be your tropic hormones. Autocrine, auto is same, right? So secretion of hormone by the same tissue that it targets. Para is what? Around, so it works on neighboring cells. All right, blood. What's in plasma? Water, respiratory gases, small amounts, ions, particularly bicarbonate ions that can help control pH balance, nutrients, waste, hormones, plasma proteins. Your formed elements are what? red blood cells, white blood cells, platelets, red blood cells carry oxygen as oxyhemoglobin. The white blood cells are for protection, so they help fight off pathogens. Platelets help with blood clotting. Foreign elements are 45% of the blood. Average plasma is 55%. What is hemoglobin?
It's the, like, hemoglobin itself, that like the red blood cell.
Hemoglobin is what's in the red blood cells that carries the oxygen. So 4 oxygen molecules are bound to the 4 iron, and that's called oxyhemoglobin, right? What is it called if carbon dioxide is bound?
Bicarbonate.
Carboxy hemoglobin, but the majority of the CO2 is traveling as what?
Bicarbonate.
Bicarbonate
Okay.
ions. Do remember that.
I was on the right track, I was like, wow, I'm
blood.
wrong.
What is a buffer?
A
So-
buffer.
Something that helps to maintain a normal pH, so it's gonna bind hydrogen ions. What is hemostasis? Stop bleeding. Stay. Hemostasis. And that is from a vascular spasm and platelet plug, which causes coagulation. Blood typing. Yeah. Please look back over that. Okay?
That's
If
not
you
right.
are negative, you can receive what? Only negative. If you are negative, you can only receive negative. If you're positive, you can receive positive and negative. Okay? That's very important. Remember that negative mom getting pregnant with a positive baby, and if she's exposed to that positive blood, her body can produce the antibodies to it that can fight off the next baby that is positive, right? So they give the RhoGAM shot to prevent the production of those antibodies by the negative mom. If you are A, you can receive what? A and O. Can you receive B? No, because you have the anti-B antibodies in the plasma. If you are B, you can receive B and O. Can you receive A? No, because you have the anti-A antibodies in the plasma. If you're AB, you can receive A, B, and O. You're the universal recipient, okay, because you don't have any antibodies in the plasma to fight anything else off. If you are O, you can only receive O, okay? You're the universal donor because there are no antigens on the red blood cell surface, so nothing can fight that off, okay? But if you're O, you can only receive O because you have anti-A and anti-B antibodies in the plasma. It would fight off anything other than O. Please go back over that, because y'all didn't get that right on your final exam. Well, most of you did. Where am I? Blood typing. Where are the antigens? Is it time to go?
No, I'm just.
Okay. Where are the antigens? Because I'm like, I'm not done. Where are the antigens? On the red blood cell surface. So if you have B antigens, you have B blood. If you have A and B antigens, you're AB. If you have no antigens, you're O. Where are the antibodies? In the plasma, and we call them anti-something. Like anti-A antibodies or anti-B antibodies, okay? Or anti-RH, but that would only be produced if you've been exposed to positive blood. Okay, if you're Rh positive, that means you have the Rh antigen on the red blood cell surface. If you're negative, you do not. Okay, what's the universal donor? O, specifically O negative. Okay, what is the universal recipient? AB positive, because positive can get anything and AB can get anything. All right, the parts of the heart, what are the receiving chambers?
Yeah.
Atrium, right? What are the pumping out chambers?
Ventricle.
The ventricles. Let's do the blood flow. So the heart comes to the heart via the superior and inferior vena cava into the right atrium through the tricuspid valve into the
Right ventricle.
right ventricle up through the
Semilunar.
similar
Semilunar.
pulmonary semilunar valve up pulmonary trunk that splits into the
Pulmonary. Right. Artery.
right and left pulmonary arteries because they're going away from the heart to the lungs for
Gas exchange.
gas exchange. How kind of blood do we have?
Oxygenated.
Oxygenated. That's gonna come back to the heart via the
Left.
two right and two left pulmonary
Veins.
because they're coming to the heart into the left atrium through the bicuspid valve into the left ventricle up through
Semilunar.
aortic semilunar valve up ascending aorta, arch aorta, descending to the whole body for gas exchange. Now what kind of blood do we have?
Deoxygenated.
Deoxygenated. That's going to come back to the heart via the superior and inferior vena cava. What is the circuit that goes to the lungs called? Pulmonary. What is the circuit that goes to the rest of the body? Systemic. Let's see. What side pumps to the lungs? Right. What side pumps to the body?
The left.
Left. That's why the left ventricle wall is so thick because it has to pump all over the whole body. What is hypertension?
High blood
High blood
pressure.
pressure.
Over what?
130 systolic.
That's average. That's not hyper.
Like 140.
Oh, you said 180. I'm sorry.
I was about to say,
No,
that's
that is
average.
way too high. That's stroke level. I'm sorry. I was thinking over 80 when you said. Anything over 140 over 90 is hypertension. What is hypo?
Low.
Low, below
90 systolic.
one.
100.
No. Oh, no. 110 over 70. But 90 is really low. Yeah.
Mine usually sits around 110.
All right, the parts of the EKG. The
Oh man.
first one, P wave is atrial depolarization. Okay, and that is from the SA node firing. Okay, and then the QRS complex is ventricular depolarization and contraction. Okay, the T wave is ventricular repolarization, which is relaxation before it starts all over again with the atria contracting with another P wave, right? So the intrinsic system of the heart, SA node, and then what? AV node, and then bundle of his, then bundle of branches, and then Purkinje fibers come up the walls. That's the intrinsic conduction system. Okay, the lungs. What is the flow? Nose, nasal cavity, trachea, pharynx, the trachea, then the primary bronchi, secondary, tertiary, bronchioles, and then respiratory bronchioles. That is the end of the, no, not respiratory, terminal bronchioles is the end of the conducting zone. Then the respiratory zone starts with respiratory bronchioles, alveolar ducts, alveolar sacs, and individual alveoli, which is where the gas exchange occurs between the alveoli and the capillaries surrounding them. Both of them are simple squamous, so that is simple diffusion. The oxygen goes from the alveoli into